Hello, everyone. I welcome you to this online lecture on carbohydrates. So, let us first start with the introduction portion. So, carbohydrates usually they are present around us. They are widely distributed around us. They are widely present in plants. The example is cellulose. They are also present in animals. And if we see the elemental composition, it is composed of carbon, oxygen, and hydrogen. But there are some other uh, elements also which are found in carbohydrates. They are nitrogen, they are the phosphorus, they are the sulfur. The chemical formula, it goes like CH2ON, and we know that all these carbohydrates, they are synthesized by plants with the help of photosynthesis. So plants are autotrophic, they produce their own food, but yes, we are dependent on the plant. This is very basic thing actually. Okay, so when we define the carbohydrates, they may be defined as the aldehyde or ketone derivatives of polyhydroxy alcohols. That means they will contain aldehyde group, they will contain ketone group, plus poly means many OH group. They will be containing many OH group. Or there are certain compounds that will yield these products after hydrolysis. They will also come under the carbohydrates. So let us first understand the functions of carbohydrates, what the functions of carbohydrates are. So first of all, we must know that these carbohydrates, they serve as the source of energy for living beings. Yes, they are the source of energy and they are termed as the instant source of energy. We say that they are the instant source of energy. Why instant source of energy? Because whenever we take carbohydrates in diet, within 2 hours, all the carbohydrates, they are absorbed into the blood and they provide energy. Energy in biological system, we know it is always in the form of ATP. All right. So moving on to the next point, they serve as the storage form of energy, which is the glycogen. Yes, glycogen is stored in the liver. It is the stored form of carbohydrate. It is composed of multiple subunits of glucose together linked by alpha 1,4 linkage. We will get to it later on. We will be studying the structure of glycogen. But yes, in the liver, the glycogen is stored. And whenever we require energy during fasting, okay, when the dietary glucose is not sufficient during fasting, during very unfortunate circumstances when we are not getting the food, this glycogen will be broken down into glucose. This will be broken down into glucose, and this glucose will be transported to the organs or to the place which exclusively survives on the glucose. See, there are organs such as brain, the testes, there are cells such as the RBCs. They are completely going to, feed on glucose. Exclusively, they will feed on glucose. So whenever the glucose is absent in the diet or we are in starvation for prolonged period of time, so what happens? This glycogen, which is stored in the liver, it is broken down into glucose. Then the glucose goes to the brain, to the different places, which exclusively need glucose as a fuel. So yes, this is a point. Next, we know that we have the non-digestible form of glucose. The non-digestible form of glucose is the cellulose, which function as the dietary fiber, which function as the dietary fiber. Now, what is the function of dietary fiber? First of all, the functions of dietary fiber is that it controls the bowel movements, the bowel movements and helps the body to get rid out of the waste. Okay, as we know, we cannot digest the cellulose. Why? Because cellulose, if we move on to the structure of cellulose, we have two glucose subunits linked by beta 1,4 linkage. In this, the glucose subunits, they are linked by beta linkages. This linkage is what? Beta 1,4 linkage. Now, so as to break this beta 1,4 linkage, we need to have beta amylases. We need to have beta amylases, and beta amylases are not present in the human beings, although there are certain fibers which can be digested by our intestinal bacteria. Yes, but beta amylases are present in bacteria. So the cellulose, it is passed through the digestive tract as such without being broken down. Okay, and it absorbs lots and lots of water and it helps the body to get rid of the constipation. All right, moving on to the another point. Another point is it acts as the structural components of glycosaminoglycans. Yes, in humans, the glycosaminoglycans, they are the structural components. All right. And the cellulose in plants, which we were discussing about right now, and chitin, which provides the structural rigidity in the insects. Apart from this, we see that the carbohydrates, they are also involved in the detoxification. For example, glucuronic acid is involved in detoxification to get out the toxins, to get the toxins out of the body, and they also form the part of nucleic acid. We know that RNA contains ribose sugar, which is a pentose sugar, and the DNA contains deoxyribose sugar, primarily 2' deoxyribose sugar. Now, what does that 2' means? That means at the second position, the oxygen is absent in the ribose sugar, so it becomes the deoxyribose sugar. We will be discussing the structures of various sugar in the next topic. All right. So let us classify the carbohydrates. So when we classify the carbohydrates, they are classified into monosaccharide, oligosaccharide, and polysaccharide. Okay. So in mono means single subunit. This is the simplest group of carbohydrates. Usually, see, whenever the plant is going to produce its own food, it is going to produce the glucose. But yes, the storage form of plants, it is the starch. What is starch? Starch is the polymer of glucose. Starch is the polymer of glucose. So from here we can understand that if single subunit of any sugar or carbohydrate is present, this is known as monosaccharide. Examples, glucose. Here is the example of glucose, fructose and galactose. So the general formula is CnH2ON, and we cannot break the glucose is a molecule. We cannot break the glucose into more simpler molecule than that. It is a monosaccharide. So this is the smallest unit of the carbohydrates, which cannot be broken down into more simpler forms. Similarly, the fructose and galactose, they are also monosaccharides. Now we will be understanding the structure in later slides. We will be understanding the structure in detail. All right. Now moving on to the oligosaccharide. In the oligosaccharide, when the 2 to 10 monosaccharide units, they are going to form various structures. It is known as oligosaccharide. If there are 2 monosaccharide presents, the sugar is disaccharide. If there are 3 monosaccharide presents, the sugar is trisaccharide. And depending upon the monosaccharide units, this is tetrasaccharide, pentasaccharide, etc. up to 10. Okay, up to 10 subunits. So now moving on to the polysaccharides. They are polymers of monosaccharide when up to 10 subunits are joined together. And sometimes the molecular weight is so high that they weigh up to the million. So polysaccharide can be further divided into two types. When similar type of subunits will be joined together, they will be known as homopolysaccharide. But when we say that different kinds of subunits will be joined together, okay, then these are the heteropolysaccharides. And by which bonding these subunits are being joined together? They are being joined together by glycosidic bond. We will be studying the formation of glycosidic bond in later slides. So first of all, we need to classify these sugars. Monosaccharide, single subunit, oligosaccharides, 2 to 10 subunits, polysaccharide, more than 10 subunits, and moreover, homopolysaccharides if the subunits are consist of same types of monomeric unit, and heteropolysaccharides if the subunits they are composed of different kinds. That is all for today. So far, we have covered the definition of carbohydrates. We have covered the functions of carbohydrates. We have covered the classification of carbohydrate. So we will be moving forward to the monosaccharides in the next lecture. Stay tuned and thank you so much.